These are some commercially available culture media and the glassware that we usually use in the lab to prepare culture media. So on the bottle itself, on the label, you can see all the ingredients and the desired pH and the desired temperature for the sterilization. And we use an electronic balance to measure the amount of powder that is need to be added from the bottle. So first of all, we have to tare the electronic balance. So after putting the watch glass, we have to make it to 0, and then slowly we can add the powder from the bottle until we reach the exact amount that we need. So once we are done with the measurement, we can add the powder into a beaker, and we usually measure the amount of water that we need to add, distilled water, using a measuring cylinder. And first of all, we have to wash the remnants on the watch glass, and then we can add the whole amount of water, distilled water, into the beaker and stir it a bit. And then you can add a magnetic bar into the beaker and place it on the magnetic stirrer. So the purpose of using the magnetic stirrer is to stir, to mix the solution well, so it becomes a homogenized solution. Once you are done with the mixing, you can adjust the pH. For that, we can use either pH papers or a pH meter. So here we are using a pH probe. So it's very sensitive, so we have to handle it with good care. And first of all, we need to wash the probe with distilled water and wipe it using a tissue, and then dip it in the solution that you want to measure the pH. So you should be careful when you dip it into the solution. The solution should not be hot, and also it should be completely immersed. The probe should be completely immersed in the solution. So if you see that there is not the desired pH, you can adjust it. So if you want to go for a higher pH, you can add diluted sodium hydroxide, or if you want to make it acidic, you can add few drops of diluted hydrochloric acid. And then you can turn on the hot plate and you can boil the solution. So once you get a clear liquid solution, you're done, and you can transfer it into an Erlenmeyer flask. So it's very important to keep a head space in the flask, because otherwise it can be dangerous, because the next step is the sterilization process. So you're going to put it into the autoclave for sterilization. So if you don't keep a headspace, there's a possibility for cracking or explosions. So then you have to plug it with a cotton wool plug and cover the top with an aluminium foil and fill the autoclave with the necessary amount of water. The coil should be covered with the water, and put it into the inside rack and seal it properly. Tighten the screw, and the operating conditions are usually 121 Celsius temperature for about 15 to 30 minutes. That depends on the type of culture medium. So it reaches a very high temperature during that period. Once it is done, so you have to wait until the pressure drops down to 0, and using protective gloves, you have to take out this autoclaved stuff. And then initially the culture medium in the flask might be very hot, so you have to let it for a few minutes to cool it down, and then you can pour it into glass petri dishes to make agar plates. So if you're doing this step inside a laminar flow, you do not have to flame the mouth every time you open it. But if you are not doing it in a laminar flow, it's important to flame the mouth always when you open it and close it, in order to avoid airborne contaminations. So this is how we prepare agar media in tubes. So you have to add molten agar before it gets solidified into tubes. So you should be very careful when you do this pouring, because you should not let it drop below 40-degree temperature, because below that temperature agar tends to solidify. And this is how we prepare slants. So initially you have to add a considerable amount of agar into a test tube, and then you have to keep it at an angled position like this. So once it is solidified, you will have a slant, a slope inside your tube.